relatively easy lecture. And you'll get the all the benefits of being here to be able to ask questions. Okay, so we'll get started. A couple of interesting things. So next week we will be taking our unknowns and we will be doing our tests on them. So if you had a gram-positive slant, you're going to start out the day doing a catalase test. If you have a gram-negative slant, you're just going to go straight into the microgen test. So you'll suspend it in your liquid, and then you guys will pipette it into the wells, put in the appropriate oil in the appropriate wells, and then you guys will be good to go. The gram-positive, if you have catalase positive, you're going to do a staph set of tests, even though they're not all staph. You'll do a mannitol salt agar, a DNase plate, and a mannitol salt agar plate, a DNase plate, and Mueller-Hinton plate. And for the strep test, you'll do a blood agar plate, a BEA slant, and a pyre broth for both of them. Now, most important thing to do before you guys do anything else is you go up to talk to TA and be like, hey, TA, this is my organism. It is a gram-positive, catalase-positive organism, or it's a gram-positive, catalase-negative organism, or it's a gram-negative organism, and they will tell you whether or not that's correct before you do anything else. Because if you grab the wrong media, let's say you're like, hey, mine is catalase-negative, so you grab this media, that means somebody else doesn't have access to this media that they need to do the tests, which would be bad. So please make sure that you guys double check with your TAs before you guys do anything. Okay? All right. So how do we identify them? Well, we've got a new improved gram-positive chart. A little bit more accurate with a little bit more information so that you guys can be able to identify it properly. And we're going to go over some of the tests that are the most important to be able to identify them. The first one, most important one, is mannitol for the catalase positives. It's not just growth, because they all grow, but mannitol fermentation only happens on 3 of them. The most important one is Staphylococcus aureus because that's really easy to tell. It's kind of got this bright, in and of itself, Staph aureus, like growing on regular media, is just kind of this light yellow color. But it creates a really nice bright yellow color on the mannitol salt agar. It's, of course, got that staphylo arrangement. And the other real key part about it is that it's susceptible to novobiocin, which is the antibiotic. Now, aside from that test, you've also got growth characteristics, which help you identify them. So if you had like a catalase or a mannitol negative one, it's kind of these 3 organisms that you'll be looking at. The biggest issue with these ones is that you can look at the color characteristics of them, but you have to do it on the mannitol salt, not on the mannitol salt agar plate. You can't do it on dye-embedded media because it's going to alter the color of the bacteria, which I think was a problem that some of you guys ran into the previous week when you were doing this. You have to look at the Mueller-Hinton agar plate or your TSA slants to be able to do that part. So, if you look at it and the bacteria is this light pink or bright yellow, it will be Micrococcus roseus, or in turn Micrococcus luteus if it's bright yellow. If it's a white coloration, and it's also susceptible to novobiocin, then it's probably going to be Staphylococcus epidermidis. Now, these 2 also have kind of a unique characteristic to them. They have tetrad formation, which is a little grouping of 4 dots, 4 foci on the microscope. So if you guys were looking at your microscope and you're like, hey, I saw this tetrad pattern repeating over and over again, this pattern of 4, then it's going to be one of those 2 organisms. So it's actually really easy to differentiate them from everything else. So that's the benefit of it. Now the hard ones are, of course, Staphylococcus xylosis and Saprophyticus. The big difference between them, aside from kind of their growth pattern, is one is going to be pyre positive, the other is going to be pyre negative. It's just kind of an obnoxious organism, but be very careful with your pyre broth. Don't add too much of the broth, don't add too much of the reagent to the paper, just a little drop of both, and then you guys should be good to be able to get good analysis of it. Now, for the catalase negative, the most important test, the easiest one to recognize is the escalin. Escalin doesn't, as long as it's really hard to mess it up or do it improperly. Basically, the bacteria are going to work or they're not. The one that's kind of escalin doesn't look super positive, but it's a little positive, a little light, is this guy, C. epidemicus, but it does show up as positive. So these 2 show up these nice, strong, dark colorations, so they're pretty easy to identify. Streptococcus agalacti and Enterococcus faecalis. I'm sorry. Streptococcus galeoptis and Enterococcus faecalis both are escalin positive, so they're really easy to identify. Differentiation, of course, with Enterococcus faecalis is that it's going to be pi or positive, whereas those are all negative. The next important one that's a little bit tricky is hemolysis. The only one that's going to be beta hemolysis for our purposes is going to be Streptococcus pyogenes. Now, the longer they sit in there, the more they can get a little bit more looking like they're in that beta thing. Particularly this guy, Zoo epidemicus. If it sits there, sometimes it'll look a little beta in coloration, but basically it should be alpha. So if it's kind of like on the border, just be like, it's probably alpha, it's probably this guy. The one that doesn't do any, that's gamma, is this guy, Streptococcus mutans. So if you got a gamma one, it's going to be that guy. And these other three are all alpha. And Streptococcus has kind of this variability growth when it comes to the antibiotic it's used with. But those are all kind of easy to kind of more or less differentiate. They've got key things to them that differentiate them, both the esculent and the alpha, beta, gamma hemolysis. Gram negatives, a little bit more complicated. We're talking about these in just a second. Most important thing to do first is the TSI. TSI is by far the most important one. It's very robust and easy to distinguish. The most important part of the TSI is the hydrogen sulfide. There are only two hydrogen sulfide organisms that we have in class, Proteus mirabilis and Proteus vulgaris. That's it. So if you got H2S positive, that's the only two. Now, some people will confuse this guy, Pseudomonas aeruginosa. It is an oxidative organism, so it cannot produce hydrogen sulfide, but it does produce kind of this black film on the surface, and people go, oh, that's H2S positive, and it trips them up. So it's a red over red slant, K/K slant, but it's got that kind of dark coloration on the slant, and they go, oh, that's positive. No, it's just the coloration of the bacteria. It's not positive. If it were positive for H2S, you could hold the slant up to the light and it would block out the light wherever that precipitate is, and the precipitate kind of sinks down into the bottom of the tube. So just keep that in mind. The other interesting ones here are our oxidative organisms. There's three of them. Pseudomonas aeruginosa, which we already talked about, Moraxella cataralis, and Alcaligenes faecalis. And those should be red over red with the exception of Alcaligenes. The reason why is a lot of people notice this. They see a little bit of yellow on the top of the slant and they go, oh, that's positive. It's not, it just sometimes creates that coloration. So just be aware that if you see just like a little bit of yellow, it might just be K/K, and it just is kind of tricking you up. But we'll talk more about how to differentiate it more thoroughly in just a second. Okay. So the other two are these guys, Pseudomonas and Moraxella cataralis, that are K/K, and they very consistently do that, so they're all red. And we're going to talk about how to differentiate those. Now, that leaves us with a bunch of other options for like Citrobacter, E. coli, Klebsiella, and Serratia. We didn't use Citrobacter. That's not in your unknown list, so don't worry about that one. You can just ignore that. It's not going to be relevant. So that one's not an option. E. coli, Klebsiella, and Serratia are an option. And the way we differentiate those, oops, jumped ahead too far. If you guys look on your test setup, the test setup does a pretty good job of identifying them. The real big one is for E. coli. It's going to be urease negative. Klebsiella will be urease positive. And they tend to be very similar to one another all the way across. E. coli is generally citrate negative and TDA negative, but that's a little bit harder to read. But they tend to be very similar in a lot of other aspects. But urease is kind of like the big one. So if it looks a lot like those two and you have the urease one, that's the one that's going to be the big clincher to differentiate it. Other important things. If you guys look at these, the sugars here, and you'll see your oxidative organisms, the ones that can't ferment, they're going to be negative all the way across for these sugars. So you can see alcaligenes, negative for glucose, mannose, xylose. Sometimes it kind of shows up a little weird on the ONPG. Don't worry about that too much. Moraxella, negative, 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 negative. Pseudomonas aeruginosa, negative, 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 negative. Easy test. So that's another reinforcing clue. Okay. By the way, if you are tripped up about the Pseudomonas and you're like, hey, there's some black stuff, I'm not sure, you do have an H2S test that works really well in your test strips, so just make use of that. So if you look here, Pseudomonas aeruginosa shows up negative on that H2S there. Okay. Well, basically, you just kind of have to work your way through and try to figure out the best one. There is one last set of things that you can use to help you identify, which is specific characteristics of these. The easiest ones are the color ones. Serratia marcescens, bright red colonies. So if your bacteria has bright red colonies, that's going to be Serratia. Pretty easy to identify. If your bacteria has nice green dye on the agar, so it's got a green coloration, just innately, that's going to be Pseudomonas aeruginosa. There is only one cosi in this entire group, and that is Moraxella cataralis. If you have a cosi from your gram staining, or a gram-negative cosi, it's that one. That's the only one. Now, some people do get tripped up, though, and I will point this out to you. Alkylates spicalis has very, very small bacilli. They're not perfect spheres. They're bacilli, but they're really tiny, and so people confuse them with cosi. If they're not perfect spheres, they're not cosi. So if you guys remember back from your slides, and you're like, it's a little squished, it's not perfect, that's probably alkylates. The last two that are kind of really easy to identify are Proteus mirabilis and Proteus vulgaris. Now we talked a little bit about the way they look. They spread on plates and slants really far, and they create kind of this film because they're very highly motile, especially Proteus mirabilis. It'll basically cover an entire plate with bacteria like that. So if you look on your slant or anything that you've cultured with your unknown, and it's spread throughout the entire thing really quickly, that would be a great example of it. Proteus vulgaris also spreads somewhat, but much less than Proteus mirabilis. Now, if you notice, there's like 3 other organisms on here. They don't have any special characteristics. They kind of overlap, which is E. coli, Klebsiella, Morganella. They all kind of are tannish bacteria. Klebsiella is technically white, and it makes this kind of like wide growth that's very mucoid, but that's not very easy for you guys to kind of look at and go, oh, it's this. Those are the ones that you're going to have to rely on the biochemical tests. Those biochemical tests are very good at helping you to differentiate them, and I'll give you guys a great example of that. Oops, jumped back too far. So Proteus mirabilis and Proteus vulgaris both spread out a lot, which we talked about before. They're both H2S positive, but the differentiation between them is Proteus mirabilis only ferments glucose, Proteus vulgaris only ferments Proteus vulgaris ferments both lactose and glucose, so it's going to be an A/A. For something like E. coli and Klebsiella, they are very similar. Remember, I talked about that before. They're differentiated by the urease. Morganella only ferments glucose, so it's only going to have a red slant and a yellow butt. So only ferments glucose. So that's kind of an easy one to differentiate if it's kind of similar to those three. So, okay. So what are you guys going to be doing? You guys are going to come in. You're going to come in. You're going to get your stuff. You're going to check with your TA that you have the correct gram organism, so gram positive. If it's gram positive, you do a catalase test, make sure that you have the right catalase result. If it's catalase positive, you're going to get these tests. If you're catalase negative, you're going to do these tests. And if you are gram negative, you're going to do a microgen test. You guys don't have to do the rapid ID ones, just do the microgen ones. That's for the 4102 class. Okay. You guys will inoculate them, you guys will put them in the incubator, and then you guys will read them the next day and do your identification. The identification is an individual task. You can ask your TA for like maybe a little bit of help or clarity, but they're probably going to refuse most of the things. This is something that you just identify on your own. Be brave, take your best guess. It's not the end of the world if you get it a little bit wrong, but you should try to do your best to identify it off of its test results. Okay, does anybody have any questions on that so far? Makes sense. Okay, last bit, cool science thing of the week. So it'll be short, and then you guys can leave, hopefully before it starts raining again. I don't think it's going to rain anymore, but maybe it will.
Later today.
Huh?
Later tonight.
Later tonight? Okay, well, you guys will get done anyways here in just a second. All right, we're going to talk about cool science thing of the week, which is actually something that was really, really recent, like within the last 2 months, which is the study involving uropathogenic E. coli. So E. coli is a source of a lot of urinary tract infections, usually the more mild ones that aren't super serious, but it's something like 160 or 190 million UTIs a year caused by E. coli, so it's a pretty good chunk. But one of the things that we found out specifically is that it alters signaling and metabolism in our cells to help them infect better. We're going to talk about that because that's really cool. So, from the authors, despite being a facultative anaerobe, we know that oxygen plays a central role in the ability of UPEC, which is uropathogenic E. coli, to colonize bladder and form stress-tolerant biofilms in the urinary tract. So the biofilms help them survive and latch on and create a persistent infection. Although E. coli encodes 3 respiratory oxidases capable of mediating aerobic respiration, a heme copper oxidase, a cytochrome oxidase BO, and 2 BD type oxidases. Cytochrome BD and BD2, only the loss of cytochrome BD impacts bacterial fitness in the bladder. So it's got 3 ways of using oxygen to drive its metabolism, and even though it's fully capable of surviving in an anaerobic environment, if it's going to infect the bladder, it absolutely needs the ability to use oxygen, and it actually only has to have one specific enzyme that makes it able to survive, which is the cytochrome BD. So if it doesn't have that one, it starts running into huge problems. So here we see 2 bacteria in, these are the bladder cells of mice. They're stained purple with actin, so you can just kind of see the outline of the cells. The bacteria you can see growing have been stained green, and the ones on the top here are the wild type. You can see they form these nice tight clusters here and here, so they're forming these biofilms. They're really close together. That gives them a really good chance of surviving any kind of immune response, so they're infecting very well. The ones that have mutations for cytochrome BD are here. And you can see that they're reduced. They're spread out. They don't have those strong clumps, and it's a very significant impact. In fact, even with the other studies, they showed the wild type infection, so they just picked mice that got an infection. They're just existing as mice. They had a much longer time to clear the infection than those that were given an infection with this muted version that's lacking cytochrome BD. So that was weird. So they're like, well, why is it doing that? You wouldn't think one gene would be this important when it's got 3 separate genes for it and 2 versions of one of them. So what they found? So they found a lot of information. The key finding that they found is the oxygen affinity of cytochrome BD is approximately 1,000-fold higher than that of cytochrome BO, which is other type of cytochrome, and intracellular human cytochrome C oxidase, suggesting that cytochrome BD promotes intracellular pathogenesis by allowing the UPEC to scavenge oxygen in the hypoxic cytosol. What does that mean? It means that it is taking away so much oxygen that is messing up the mitochondria within these cells so that they are not able to survive and do their normal function. It's altering their metabolism because it's taking away all of their oxygen. There's no free oxygen. Well, it's not no free oxygen. There's limited free oxygen for them. On top of that, as they increase that consumption, it depletes cytosolic oxygen, which stabilizes hypoxia-induced transcription factor, HIF1, which induces a shift towards aerobic glycolysis. So it changes the way the bacteria handle their nutrients. It changes the way bacteria are able to utilize nutrients. It's also altering this transcription factor, which we're going to talk about in a second. Circle back to that. So what does this look like in a cell? So they have an example here. They've got a normal bladder cells from mice. They've got the blue part is DAPI stain, so that's just the cells. The purple one is the mitochondrial parts within those cells. And then you can see those little green guys there. Those are the bacteria. And you can see the top one, the control one with no bacteria. There's no bacteria in there. The mitochondria is evenly spread throughout the entire thing. That's what we expect to see. The one that's got the urinary tract infection bacteria, you can see the bacteria in there, and that the mitochondria have reduced significantly. They talk about the surface area of them. You see here that this scale's a little fuzzy, but basically this is talking about how much mitochondria there is. It's about 20 micrometers squared right here, 20 to 25, 30. But here, it's actually dropped down to 2. That's a pretty big drop. So that's interesting. So they're altering the way the mitochondria are existing. They're messing up the mitochondria. These guys, though, that have the mutation, they have much higher mitochondria surface area. It's statistically significantly higher, even though it doesn't look like it much. It's, instead of being 2, it's up to 2.5, 3. But still, that's much better. But if you also notice, the spread of the mitochondria is a lot more intact on the BD mutation one. So the ones that don't have the functioning cytochrome oxidase, they don't seem to affect the mitochondria as much. On top of that, we were talking about that signaling thing. That signaling protein actually tells bladder cells, hey, you know what, you've been around too long, you probably need to die off and slough off so we can put in new cells. Because bladder cells take a lot of damage from being in a very caustic environment, so your body sloughs off urinary bladder cells quite regularly. However, if you're in a bacteria and you want to latch on to someplace and infect it, you don't want to be accidentally sloughed off with those cells. So they've been signaling and altering the signaling pattern so that these cells are not accidentally killing themselves off, apoptosis, to slough off and allow new growth. They're keeping these cells alive and basically holding them off from dying, specifically through creating an oxygen-deprived environment, which is a key trigger for that type of apoptosis, and by altering its signal pathways. So, I know, very complicated thing. What does this mean? Well, it offers us two really important treatment targets for drug manufacturers, and all you have to take away from this is one. These bacteria are heavily reliant on oxygen metabolism, even though they're really well known for their anaerobic metabolism, E. coli are, to cause bladder infections. And the way they use that aerobic environment is to alter the way mitochondria and cell signaling happens in our cells, which is crazy. They are actually not just kind of, defending themselves away from our immune system. They are literally changing the way our body signals to itself and how our body does metabolism. So it's actually really crazy. All right, so that's all you have to take away from it. Does anybody have any questions? Okay, I will let you guys go. Have a good weekend. If you guys have any questions, let me know. And good luck on your unknowns next week. Oh, and there is no quiz this week. So just chill, you don't have to take a quiz. It's all stuff you know already. Unless you guys want to take a quiz on this school science thing of the week.